Spike protein virus genetics and mutation. Please refer to the previously posted constituent factors in viral diseases, parts 1 through 5, for background data relevant to this presentation. Mutation consists of a permanent transmissible change in genetic material, generally involving a single gene in a cell, virus, or organism, including gross alterations in chromosomal structure. Virus mutations occur due to natural selection, random genetic drift, or epidemiological factors affecting a population. Natural selection refers to the survival of individual entities and their progeny that become best equipped to adapt to environmental conditions. Some viruses are capable of adapting to novel hosts and environments as a function of their ability to generate de novo diversity in a timely manner conducive to their further survival. Spontaneous mutation occurs in all organisms, attributable to their inherent rates of replication error as well as genomic transmission. RNA viruses mutate more rapidly than DNA viruses. Single-stranded viruses mutate quicker than double-stranded viruses. The degree of viral mutation evolves in response to particular selective pressures. Various virus and host-dependent processes are determinants of viral genetic diversity. The evolution of viral mutation rates occur as a matter of natural selection. The genetic mutation rate consists of the probability of a change in genetic information being passed on to the next generation. A viral cell infection cycle involves its attachment to the host cell surface, entry, gene expression, replication, and encapsulation, resulting in the release of new infectious particles. Mutations are random events. Viral mutations involve DNA mismatches by proofreading or post-replicative repair. Host enzyme activity, spontaneous nucleic acid damage, and the genomic elements specifically productive of novel mutations. Viral genetic mutation is also modulated by replication proteins other than those associated with polymerase, replication mode, as well as template sequence and structure. A genetic template is a DNA or RNA strand synthesized in replication or translation, resulting in a new synthesized strand complementary to the template. Mutation is a biochemical and genetic process. The Baltimore classification of 7 virus types is based on the genetic material comprising the virion: positive strand RNA viruses, negative strand RNA viruses, double strand RNA viruses, retroviruses, pararetroviruses, single-stranded DNA viruses, and double-stranded DNA viruses comprise this classification. Coronaviruses are enveloped single-stranded positive sense RNA viruses, sense, positive sense, and antisense, negative sense, refer to the 2 strands of double-stranded DNA, which are the template for transcription. The sense strand contains the specific nucleotide sequence to the messenger RNA, encoding for a functional protein. Antisense is the template for transcription, containing the complementary nucleotide sequence to the transcribed messenger RNA, functioning in protein translation. The sense strand cannot be transcribed into mRNA. The antisense strand is the template for translation. One strand of a double-strand DNA molecule is the negative sense strand, complementary to its positive sense strand, as well as the RNA transcript. Coronaviruses are a family of positive strand RNA viruses having a proofreading capability as opposed to the other RNA viruses. Genetic proofreading is an error-correcting repair mechanism of the DNA replication process catalyzed by DNA polymerase. An exonuclease is an enzyme removing successive nucleotides from the end of a polynucleotide molecule. This exonuclease activity is believed to provide for the coronavirus proofreading function of RNA polymerase and its ability to maintain a large genome size. Polymerase fidelity is the incorporation of correct bases and the exclusion of incorrect bases from the active site during DNA synthesis, functioning as the main mutation rate determinant. Fidelity variants provide for the ability of RNA viruses, like the coronavirus, to adjust mutation rates on an evolutionary basis in response to natural selection pressures acting on the mutation rate or other viral characteristics. This provides for the ability of coronaviruses to rapidly mutate into novel infectious forms, perpetuating its ability to survive against selective pressures, infect new hosts, and remain viable. Coronaviruses are single-stranded viruses consisting of 30,000 bases, comprising the longest genome of any RNA virus. Protein kinases add a phosphorylation marker to other specific proteins, determining their function. Viruses utilize the biochemical machinery of host cells to infect, reproduce, and spread. Viruses are capable of controlling the function of specific kinases to overtake and specifically control the cellular machinery of the infected host for this purpose, also resulting in corresponding signs, symptoms, morbidity, and mortality observed in coronavirus patients. Protein kinase R is an important effector of innate antiviral immune system functions, such as apoptosis, also referred to as programmed cell death, occurring as part of an organism's normal growth and development. Genetic selection causes viruses to exhibit continuous, subtle, and extensive genetic changes by means of mutation and recombination, respectively. Mutation takes place due to the incorporation of an error in the genome of the virus. Recombination results if co-infecting viruses exchange genetic information and a novel virus is created. RNA viruses exhibit higher mutation rates on the order of about 1 per virus genome copy compared to that of DNA viruses. Mutations may have harmful, neutral, or positive effects. Mutations not interfering with essential viral function are capable of persisting in a given virus population. Mutations may produce viruses exhibiting novel antigenic determinants in a process referred to as antigenic drift. Such viruses resulting from antigenic alteration may cause disease in formerly resistant or immune hosts. Mutations can result in viruses exhibiting altered host range or altered target cell specificity with intact antigenicity. These viruses may be useful in the development of vaccine strains. Recombination resulting from independent gene assortment may take place among viruses possessing segmented genomes. These open reading frame genomes, common to RNA viruses, facilitate genetic reassortment among different strains in the same cell possessing a source of viral variation. The open reading frame is a sequence of nucleotides in a nucleic acid transcribed into RNA, whereby the ribosome can transit adding consecutive, non-overlapping amino acid triplets before before it encounters a stop codon, no longer coding for any additional amino acid during translation, ending the open reading frame. A codon is a sequence of 3 nucleotides forming a unit of genetic code in a molecule of DNA or RNA. DNA consists of repetitions of ACTG nucleobases, providing for transcription into RNA, subsequently translated into protein. Messenger RNA is then read not 1, but 3 letters at a time, as a codon that is interpreted by a ribosome functioning as the molecular machine productive of a particular amino acid. A codon on an open reading frame is 3 nucleic acids in length. In DNA having 2 strands, the ribosome is capable of reading RNA derived from one strand or another. It is read in separated tripartite units, resulting in 3 reading frames reading in one direction as well as 3 in the other direction. 6 different reading frames for each DNA may produce an open reading frame. Genes residing on different nucleic acid pieces are randomly assorted, resulting in viruses having novel antigenic determinants and host ranges. Antigenic shift involves the development of viruses possessing novel antigenic determinants by means of independent assortment. Genes on the same nucleic acid may experience recombination. The closer 2 genes may be, the more remote is the possibility of their recombination, in this instance of partial linkage. Novel virus development with novel antigenic determinants, regardless of the means of recombination, may provide for infectious viruses causative of disease in formerly immune hosts. Viral vaccination results in the replicating virus expressing a specific immunogenicity, stimulating antibody production with specificity, protecting the host from the immunogen and the vaccine virus. Viral strain reappearance may result from recombination, involving independent gene assortment from 2 different viruses. Antigenic shifts can lead to pandemic disease. Viral recombination consists of viruses from 2 different parental strains affecting a given host cell, interacting during replication, generating viral progeny possessing genes from both parents. This usually occurs between the same virus types. One recombination mechanism is independent assortment of viruses possessing segmented genomes, trading segments during replication whose genes are unlinked and randomly assorted. Recombination can be between genes located on the same piece of nucleic acid. Another form of recombination is incomplete linkage. Genes usually segregating together are linked genes. Recombination occurring between these genes involves incomplete linkage. Recombination of incompletely linked genes has been observed in DNA and RNA viruses. Recombination occurs in some positive sense single-stranded RNA viruses, such as the coronavirus. Coronavirus recombination occurs at the point of viral RNA genome interaction by means of a copy choice mechanism. This occurs because the viral RNA polymerase binds to solely a few bases of template RNA at a time. This polymerase template RNA interaction persists, causing the polymerase bearing the RNA strand to dissociate from the template nucleic acid strand and associate with a novel template RNA strand, in this gene combination process. Evolutionary variation involves differences between parents as well as their offspring, or between individuals in a population. Genetic variation consists of mutation, a process productive of a gene or set of chromosomes differing from the wild type. Recombination, a process in a diploid or partially diploid cell generating a novel gene or gene combinations not found in that cell or its progenitors. Immigration, movement of genes resulting in an alternative gene type or allele. Gene mutations occur via the effects of physical mutagens on nucleic acids, the natural base functions of nucleic acids, and the failure rate of the enzymes of nucleic acid replication. Viral mutation rates depend upon the fidelity of their enzymes to replicate their nucleic acids. Viruses possessing high fidelity transcriptase exhibit relative low rates of mutation and vice versa. Mutations interfering with attachment, penetration, uncoating, replication, assembly, and release do not exhibit misapplication and disappear from the population. Genetic code redundancy provides for many neutral mutations and no viral protein change, or in replacement of an amino acid by one that is functionally similar. Mutations that do not impair necessary virus functions can remain or become established in a viral population. Mutations may result in novel antigenic determinants, resulting in antigenic drift, enabling a virus to prevail against host defenses, causing disease in previously immune hosts. Mutations occur at a slower rate in coronaviruses because they proofread after replication, reducing the chance for possible errors. Viruses have the tendency to result in less morbidity and mortality as herd immunity increases with less opportunities of viral spread to a novel host. Viruses are capable of initially being very virulent and causative of morbidity and mortality, then slowly mutating, resulting in increased chances for host survival in future cases. The virus may be capable of living in a dormant mode in the host, reappearing at a later time. Much more research regarding mutation, herd immunity, and the post-infection conferral of long-term immunity from prior infection or specific vaccination is essential to the understanding of epidemic and pandemic COVID-19. New presentations coming soon.